Moderators, and so this is a session on clinical trials. I'm always very excited about this session. It started a few years ago, and it's grown and gotten a lot of good feedback. I think, as we all know, clinical trials are, how we make gains in this disease, and so this is a kind of a snapshot at the most promising clinical trials that are out there. Each year we get quite a few submitted abstracts, and it's really a very difficult decision because so many of them are very good, but this year we picked, I think, 6 of them that we felt that you would be really interested in, with either novel treatment approaches, phase 3 trials with a high hope of changing standard of care, or new combination treatment. So I will turn it over to Ty here to announce the speakers, and forum, I think, will be each person will present their trial, and then I think we'll probably do questions at the end so that we make sure we get through everybody's presentation.
All right, thank you. So first, I want to introduce Dr. Dizman talking about Ivonescimab in the IVORY study.
Thanks, Dr. Ho. Good afternoon, everyone. I'll be talking about the IVORY trial. This is a phase 1 study of Ivonescimab in previously treated advanced metastatic renal cell carcinoma. Ivonescimab, as a background, is a novel first-in-class inhibitor bispecific against PD-1 and VEGF. It simultaneously blocks VEGF and PD-1. The preclinical data with Ivonescimab indicates a potential synergy with use of Ivonescimab and inhibition of PD-1 and VEGF. The promise with this agent is that the close proximity of PD-1 and VEGF inhibition and simultaneous inhibition could yield ways to overcome immune resistance in patients who received immunotherapies in the past. In terms of clinical data with this agent, the most robust clinical data comes from non-small cell lung cancer experience. In HARMONi-2 phase 3 trial, Ivonescimab demonstrated superior activity over pembrolizumab alone, and it was tolerated well. Our study is designed to assess early clinical activity signals of Ivonescimab in renal cell carcinoma. In this trial, we are going to enroll 40 patients, and they will be enrolled into 2 different cohorts. The cohort 1 will include the patients who have received immunotherapies in prior lines, but not VEGF-targeted agents. And cohort 2 will enroll the patients who have received immunotherapies and targeted therapies in prior lines of therapies. Patients will receive Ivonescimab at 20 milligrams per kilogram dose every 3 weeks in outpatient setting until disease progression or unacceptable toxicity. The primary endpoint of the trial is objective response rate and disease control rate, while the secondary endpoints include toxicity, progression-free survival, overall survival, and duration of response. We are collecting pre-treatment, on-treatment, and at progression tumor tissue, blood, and stool samples for correlative analyses. We will conduct this trial using the time to event Bayesian optimal phase 2 design. The 2 endpoints will be monitored simultaneously in each cohort separately. The null hypothesis in cohort 1 is that assumes that the objective response rate would be 20% and disease control rate would be 30%, while the alternative hypothesis assumed an objective response rate of 30% and disease control rate of 50%. With this plan, we will accept this agent as having a clinical activity if we observe objective responses in at least 4 patients and disease controls in at least 9 patients. With this plan, we will have a power of 96% to detect early clinical activity of this agent with an alpha of 20%. In cohort 2, no hypothesis assumes an objective response rate of 5% and a disease control rate of 20%, while the alternative hypothesis assumes an objective response rate of 20% and a disease control rate of 40%. We set the bar lower here, and with this plan, we will accept this agent to be active if we have at least 3 objective responses and 7 disease controls. And this design will give us 91% power to detect clinical activity signals with an alpha of 12%. For conclusion, this is the first clinical trial of bispecific against PD-1 and VEGF in renal cell carcinoma. Potential clinical activity signals and the translational insights obtained from this study will allow us to understand mechanisms to revert immunotherapy resistance in renal cell carcinoma and hopefully open new venues for this agent to be tested in other settings of RCC. I'd like to thank Dr. Msal, Dr. Tanir, and MD Anderson GU Oncology team for the opportunity to work on this trial, and KCRS for the opportunity to discuss that with you today. Thank you.
All right. Thank you very much. I wanted to invite Dr. Kotecha to talk about a novel bispecific.
Thank you so much to the organizing committee for selecting this abstract. My name is Ritesh Kotecha. I'm a medical oncologist at Memorial Sloan Kettering Cancer Center. And on behalf of my co-investigators, I'm glad to present here this trial in progress of XmAb819, a novel ENPP3 CD3 T-cell engager for patients with clear cell renal cell carcinoma. So by way of introduction, ENPP3 is a naturally occurring enzyme with high and differential expression in clear cell RCC. The expression of ENPP3 in normal human tissues is low and restricted, mostly to basophils and mast cells. XmAb819 is a humanized anti-ENPP3 CD3 bispecific antibody that directs T-cell mediated cytotoxicity. As you can see in the figure here, this has a unique 2+1 formatted design with 2 arms that bind to ENPP3 with 1 to CD3 to essentially provide avid tumor targeting and selectivity to high ENPP3 expressing cells like clear cell RCC. In this trial, this is a phase 1 multi-center first-in-human study of XmAb819 in patients with relapsed or refractory clear cell. Currently, the trial is enrolling here in multiple centers in the United States with a plan to enroll globally. As you can see, this is a dose escalation currently with both intravenous and subcutaneous formulations. Similar to other T-cell engagers, patients after enrollment are treated with a priming cycle where they are exposed to a low priming dose and essentially step up to full target doses to mitigate the risk of cytokine release syndrome. They are then treated with that full dose on subsequent cycles with an interim monitoring for anti-tumor activity. As you can see, the primary endpoints are safety and tolerability, and essentially the determination of a recommended phase 2 dose for expansion. The vision is to obviously enroll more patients in clear cell RCC in an expansion cohort to even branch out also to other solid tumors that are ENPP3 expressing. For this crowd, this includes papillary RCC and other solid tumors like colorectal cancer and non-small cell lung cancer. Secondary objectives include anti-tumor activity, and exploratory objectives are to look at pharmacodynamics and also correlates with ENPP3 expression. Thank you.
Thank you. All right. I'd like to invite Dr. McGregor to talk about the STRIKE study.
Right. Well, thank you so much for the opportunity to speak to you about the STRIKE trial. So this is a short-term intensified pembrolizumab with tivozanib for high-risk renal cell carcinoma. So as everyone knows, pembrolizumab has been approved by the FDA, and we not only see improvement in relapse-free survival, but an improvement in overall survival. And I think as you look at this data, there are 2 questions you can ask yourself. I think there's been a lot of questions about, are we over-treating patients? But at the same time, I think there are patients who still relapse despite pembrolizumab. And so we chose to answer the second question, how can we improve outcomes further, and how can we build upon the success of pembrolizumab? Tivozanib, as everyone knows, is a well-tolerated oral VEGF TKI. And I do want to point out that we have data from TiNivo-2, where that if we look at the exposure response analysis, tivozanib at full dose with nivolumab should not increase hypertension, which was the concern based on the initial analysis. So as we think about this, tivozanib at full dose should be well tolerated. And for more information on this, I would ask you to see the poster outside. So that brings us to the STRIKE design. So this is A032201, and we are really looking to build upon the success of pembrolizumab, studying the exact same patients that were studied in Keynote 564, so T2 grade 4, T3 any grade, N1, M1 NED within a year, with the caveat that we are going to be allowing patients to have ablation for their metastasis. So either SBRT or an ablation will be allowed. Looking to enroll just over 1,000 patients. Patients will be randomized to pembrolizumab for 12 months or pembrolizumab for 12 months with just a 6-month course of tivozanib at 1.34 daily, day 1 through 21, every 28 days for 6 months. Dose reductions to 0.89 every other day are allowed. There's no limits on dose interruptions. Key secondary endpoint will be overall survival. And at the same time, while we're looking to intensify, we do not want to stop there. We want to determine, use this study to answer those questions about who actually needs to be treated, what are markers of residual disease, is a host capable of responding to therapy, is a tumor capable of responding to therapy, and hopefully with the help of David Braun and others, we can collect these samples and try to answer these pivotal questions. And to that point, we're going to be having optional studies to collect blood at baseline, week 24, 12, 16, 24 months. We're collecting radiology for radiomics, and we're hopeful that this will answer some of those key questions. Where we are right now, we have 220 sites open, 614 pending. As of Friday, we had 33 patients. I looked this morning, we had 35. Dana-Farber has 7, of which 5 of them are mine. So I'm hoping that everyone else can get the same enthusiasm as I do, and we can get this study across to the finish line. As you know, there's really nothing else open right now in this space, and I hope we can help answer this important question.
All right, thank you very much. I'd like to invite Dr. Ravi to talk about radioligand therapy.
Hi, good afternoon, everyone. Thank you for the invitation to present this trial. It's called LASER, a phase 2 trial of lutetium PSMA-617 in kidney cancer. And you read that right, it's not prostate cancer. So why do we want to do this study? So clearly there's a need for new therapeutic targets in clear cell RCC and other RCC as well. PSMA is actually present on the neovasculature of many solid tumors, including clear cell RCC, and it's been confirmed in relatively small studies by IHC and RNA-seq. This is a publication using, I think, the Tempus or Keras database showing that, in particular in clear cell. And it can also be imaged by PSMA PET, as we know for prostate cancer. This is a meta-analysis of, again, relatively small numbers of patients, this is literature to date, showing that PSMA PET can identify disease in clear cell RCC in 80% of the time. And we know that there's a drug. It's a theranostic target with PSMA PET. We have lutetium PSMA-617, which has proven activity in PSMA-positive mCRPC, castrate-resistant prostate cancer. So here's the design. It's a pretty simple design. It's a single-center investigator-sponsored trial. These are all patients who've got advanced clear cell RCC, having had at least 1 prior IO and 1 prior TKI. The eligibility requires at least 1 PSMA-avid lesion with avidity greater than liver, and no PSMA-negative lesions per vision criteria, which allow lymph nodes up to 2.5 and visceral lesions up to 1 centimeter. And patients basically get 2 cycles of lutetium PSMA if they're eligible. They have an interim PET scan, and if that PET scan shows residual avid disease, they get up to 4 more cycles. There's also optional tumor biopsy at baseline and prior to cycle 3, and blood for circulating tumor DNA being collected every cycle. And the design is that we'll enroll 9 patients initially. If at least 1 RECIST response is seen, 15 more patients will be enrolled for a total N of 24. And the primary endpoint is objective response rate. And the target response rate is 25%, which is very similar to belzutifan, basically. So the vision and impact, this is really one of the first PSMA RLT studies in RCC. There are a couple of others, very similar ones ongoing, I believe, looking at clinicaltrials.gov. There's also CA9, obviously RLT, which is in the pipeline. There's a new target, and it's a new class of drug in this disease, and it can be easily combined, particularly with things like TKI and IO. We know that from prostate cancer anyway. So promising activity here could lead very easily to a large randomized study. And we have embedded imaging, tissue, blood-based correlatives to look at mechanisms of response resistance, the interplay between the immune milieu and the response, and really inform future efforts looking at RLT in this disease. Thank you.
Thank you. I'd like to invite Dr. Runchi to talk about zanzalintinib.
Just this one. Okay. Good afternoon.
Do you need to put it back on? Or you-
Yes, it's going to the wrong slide. I will.
I think just start there.
Okay.
You just start with
We'll just start. So I'll present on the EXACT study, which is a randomized phase 2 study of XL092, now known as zanzalintinib, in patients who progress on adjuvant therapy in advanced clear cell RCC. And this is a multi-center study that is being conducted through the Motzer Cancer Research Network. So the rationale for the study is that, Keynote 564 was a large phase 3 randomized double-blind control study which led to the approval of adjuvant pembrolizumab in clear cell kidney cancer. At almost 5 years of follow-up, 30% of patients who received adjuvant pembrolizumab had disease recurrence, about 15% locally and almost 89% distant recurrence. There is no optimal treatment regimen for those patients who progress on adjuvant pembrolizumab. Zanzalintinib is a novel oral multi-kinase inhibitor which has demonstrated safety, anti-tumor activity, and immunomodulatory effects in treatment-refractory clear cell kidney cancer. Phase 3 studies are ongoing. Preclinical data has suggested that zanzalintinib in combination with anti-PD-1 may be synergistic. The EXACT trial. So this is a randomized phase 2 trial. Eligible patients have confirmed clear cell kidney cancer, measurable disease as per RECIST. They've progressed on adjuvant pembrolizumab, no prior zanzalintinib, and ECOG performance of 0 to 1. Patients will be randomized 1-to-1 to either treatment arm, zanzalintinib or zanzalintinib plus nivolumab. The primary endpoint is overall response rate at 6 months. Secondary endpoints include progression-free survival, overall survival, and adverse events. Exploratory endpoints include the % change in ctDNA. We're going to look at changes in the tumor immune microenvironment, as well as radiomics and tumor kinetic analysis. The impact. Zanzalintinib is a novel drug. This is the first study in this space. So zanzalintinib plus or minus nivolumab may inform future phase 3 studies for patients who progress on adjuvant pembrolizumab. Pre- and post-treatment blood and tissue correlates may identify mechanisms of immunotherapy resistance in advanced clear cell kidney cancer. And perhaps this may shed light on the question of IO re-challenge after adjuvant pembrolizumab. I'd like to thank the patients and their families. I'd like to thank my collaborators, Hoosier, and thank you to KCRS for the opportunity to present this study.
Thank you. I'd like to invite Dr. Xu to talk about natural killer cells.
Hello, and thank you for the chance for me to present on behalf of my co-investigators on memory-like natural killer cell immune therapy in patients with kidney cancer or urothelial cancer. The background is that we know that natural killer cells have a role in anti-tumor immunity. In single-cell analyses of patients with renal cell carcinoma, which was done in collaboration with Dr. David Braun, Dr. Shueri, and Dr. Romi, we've seen that dysfunctional NK cells are enriched and are progressively more enriched as patients progress to higher stages of RCC. These dysfunctional tumor-infiltrating NK cells have impaired cytotoxicity and express tissue residency markers. In 2 independent cohorts, we looked at a gene expression signature that was derived from single-cell RNA-seq from this dysfunctional NK cell population and found that these expression signatures are associated with worse survival in kidney cancer. The Romi lab in parallel has shown that cytokine-induced memory-like NK cells have enhanced cytotoxicity, prolonged survival, and better proliferation compared to these native tumoral NK cells. And so we're asking the question of whether we can restore NK cell anti-tumor activity in patients with RCC. This is a first-in-human pilot study. In this study, patients with advanced RCC, which includes clear cell, but also chromophobe or translocation RCC, or urothelial cancer, and who have had progression on prior therapies, undergo apheresis for autologous natural killer cell collection. The natural killer cells are manufactured into cytokine-induced memory-like NK cells through a purification and cytokine stimulation and expansion process. During this time, the patients undergo lymphodepleting chemotherapy with fludarabine and cyclophosphamide, followed by infusion of their autologous memory-like NK cells. This is then followed by up to 5 doses of low-dose IL-2, and patients are followed by outcomes. The primary outcome of the study is feasibility, as defined by ability to collect cells, generate, and administer the seminal NK cells to patients, and we are also monitoring for safety and preliminary activity. 5 out of 10 patients have been enrolled to date. Overall, this is the first trial to investigate cytokine-induced memory-like NK cells in kidney cancer and urothelial cancer. We are especially interested in potential activity in these divergent histologies. Correlative studies, which are funded by the CDMRP and by the Kidney Cancer Association, with Dr. Tony Shueri and Dr. Rizwan Romi as co-PIs, will explore the phenotype and function of NK cells and determinants of treatment response, including comparison of responders versus non-responders. These data will help to inform ongoing work on NK cell-based therapies in kidney cancer. In particular, we are currently going through preclinical development of a next-generation product, which consists of an IL-12A3 fusion cytokine secreting CD70 CAR-NK. And for more information about the preclinical development of this new concept, please see the details from the poster from Dr. Liu and Dr. Deng. Thank you for your attention.
All right. Well, thank you very much. We're pretty much right on time. This is definitely a group you want to take into the Squid Games because we have like 20 seconds left. So I'll walk into the crowd and we'll take questions.
The question is regarding the, the Pluvicto trial, which I think is a brilliant idea. The question, though, I just wonder, because, kidney cancer is less sensitive to beta radiation than is prostate, almost certainly. Have you guys modeled the relative dose response, and do you think you'll get enough effective, basically effective radiation dose with the PSMA lutetium construct?
We haven't, and it's really, exploratory. If we see something, I think that'll be exciting, but it's honestly sort of more as an opportunistic study. Given that we have the drug, we have the target, we can give it safely, and we'll try and see if it works.
Good luck. Thanks. I mean, the most recent studies that we looked at earlier showed that higher doses of SBRT are required, and lutetium, the PSMA lutetium doesn't deliver that high of a dose with the. I mean, you're the radiation oncology expert. You should tell me.
Hard to correlate.
Yeah.
From the data.
Why don't we wait and get a microphone so the people in the back.
Here, I'll go throw it across.
Hard to correlate. I mean, Chad can comment on this as well in terms of what does that mean from the dosimetry, by the way, for most of the radioligand therapies is very poorly understood, is the most charitable way to phrase it. I had a slightly different question for Dr. Ravi, which was, many of the radioligand therapy trials are going to a more adaptive response, and why did you choose to have 6 doses?
So I mean, I think that's a great point. I mean, we're starting to learn how to even do adaptive dosing in prostate. I mean, the ENZA-P study has introduced that. So we kind of are doing a little bit of an adaptive dose. We're doing 2 doses, then an intrapet, and then 4 more. So we want to stop, obviously, if there's no target. But you know, this is the first time we're trying it, so I think it made sense to stick to the doses that we know about rather than do something kind of different. But I would love to integrate dosimetry, for example, in future studies. If we see activity, I think we could easily add further cohorts to this type of trial.
Can I ask the same question along the same line? Is there diagnostic approaches? PSMA has been used, diagnostic approaches for prostate cancer. Is there enough imaging data from the kidney cancer field as far as the imaging results from large patient studies?
It's not large patient studies. I think that meta-analysis I showed you was maybe 250 patients because PSMA PET has only been around 10 years in Australia. It's been around since the US for 4 years. Now, we didn't screen the first patient last week, and the patient had SUV max at 20.
Please come talk behind. He's already
disease, so it's clearly there. It's at a level where you should think it should be therapeutically targetable. We obviously won't know, but it's very limited data in terms of imaging of PSMA PET in RCC. Yes, that meta-analysis was including primary and metastatic sites, but they broke it down, and it was about similar detection rate for both primary as well as metastatic lesions.
A quick follow-up on the PSMA. So how does the PSMA expression per se evolve in adjuvant versus metastatic and after IO or IO-TKI exposure to the patients? Do we know that?
We don't know.
Okay.
Yeah, I mean, the data is very limited, right? I mean, there's very small numbers of patients who've received PSMA PET. These are small studies from IHC. They said it's typically metastatic tumor samples. So there's a lot of work to be done. I think if we see activity, then I think they'll promote a lot of work in this space and RLT in general in RCC.
Thank you.
Brad, I know this is a valiant effort to design a 1,000-patient phase 3 trial in the adjuvant setting, but there have been, we all know there have been several trials with single-agent, TKIs that have been negative. SOURCE, ASSURE, PROTECT, ATLAS. Why do you think TiVo is going to be different than the others to really launch? I understand there is no adjuvant trial right now, but do we really need to have this trial? I, for one, do not believe there is synergy between anti-PD-1 and a TKI. And I predict that the trial is going to be negative. Is it really worth the resources?
So, I mean, I appreciate the question. I mean, obviously, this has been like years.
I know the train has left the station.
From my standpoint. But I think at the end of the day, I think that IO-TKI is a standard of care in the metastatic setting. In this question of synergy versus additivity, I think people have their beliefs, but we don't know that. I think as we look at this right now, the thought is tivozanib is well tolerated. We should be able to give the full dose for the 6 months, and if there is a synergistic effect, that 6 months should be enough. And we've seen like hints of activity. If you look at the PROTECT trial, if you look at S-TRAC, like when you maintain that high dose, there was some DFS benefit. And so I think this is an important question. I think beyond the trial, we are collecting blood and radiology, so we can try to answer some of these questions in better detail.
So another comment to Carrie. I mean, we have 2 phase 3 trials that looked at PD-1 blockade after progression on a PD-1. Why the arm with Nivo plus Zanza after progression on adjuvant pembro? I mean, how does that make sense?
Well, this trial was designed before those studies came out, and I think this is a slightly
slightly different patient population. These patients don't have the primary tumor in place, which is thought to be a big source of immunosuppression. This is a novel drug that's being tested, and we don't know what the effects will be in combination with immunotherapy.
Hi, Nazli. Question, I'm very interested in your bispecific trial, and I was wondering if you think there might be less toxicity with that approach, whether you're noticing in the patients that have been treated initially, are you seeing less dysgesia or less hand-foot or any of that?
Thanks, Dr. Haas. This is something we specifically reviewed the literature for. We have recently enrolled our first studies, first patient, so we haven't treated anyone with this agent yet. But looking at the lung cancer experience, actually overall adverse event profile in terms of both treatment-related adverse events and immune-related adverse events looks very similar to what we would observe in an immunotherapy plus beg-directed treatment combination. Interestingly, though, in the lung cancer trial where they treated 200 patients with metastatic disease, only 3% of the patients needed to discontinue treatment due to any treatment-related adverse events. So we are hoping for a smooth safety course for our patients as well.
I have a question for Vincent. Now that you have these NK cells, they can kind of kill tumors that have downregulated MHC, so they're kind of more like a gamma delta than an alpha beta. Do you see evidence of MHC downregulation as a resistance mechanism in kidney cancer?
That's a great question. So the answer is yes. There is data for MHC downregulation as an immune escape to PD-1, PL-1 inhibitors in RCC. And one of the potential advantages of NK cells is that NK cells may be less susceptible to this immune mechanism, and it may actually make them more vulnerable to NK cells potentially.
Any other questions? Okay, thank you very much. Oh, one more question, sorry about that.
One question for Dr. Ravi. For some of the prostate studies, they were designed so that the majority of the lesions are actually PSMA avid. Do you think that having one lesion only to be PSMA avid may set the bar a little bit low?
So the eligibility requires at least one avid lesion and no negative lesions. Okay. So obviously the patient who's got one avid lesion, quite reasonably, they would probably go for SBRT or something like that. But these are patients post-IO, post-TKI, they're going to probably have multifocal disease.
All right, well, thank you very much.